So good morning, everyone. For me, I'm sure it's not morning for the rest of you. Sorry about that. I want to welcome you to today's joint RDA ESIP physical samples webinar. And thank you so much for taking time out of your life to be here. My name is Stephanie Hagstrom, excuse me, and I am the Director of Community Development at the Research Data Alliance US. And give you a little background about the RDA. The Research Data Alliance is a community of over 11,500 data experts from around the globe who collaborate together in building the social and technical bridges to enable open sharing and the reuse of data. The RDA US is a recognized region of the Research Data Alliance, represented by 2,500 members that support the global operations and help strengthen the data science infrastructure community. If you're not already an RDA member, we really encourage you to join, and we hope that you'll take part in this very vibrant community. We are very excited about this particular webinar series as we are partnering with ESIP on this project, and I am thrilled to turn it over to Megan Carter for a little bit about ESIP.
Hello, everyone. As Stephanie mentioned, I'm Megan Carter. I'm ESIP's Community Director, and I wanted to add that we too are very excited to kick off this joint webinar series. The Earth Science Information Partners, or ESIP, is a global community for earth science data professionals. We aim to advance the usefulness and impact of data to address global environmental challenges and to fuel new discoveries. So for more than 20 years, ESIP has helped members of the earth science data community to cross traditional boundaries, to leverage each other's expertise, and to work together on common data challenges and opportunities. ESIP fosters rich collaborative experiences, including 2 annual conferences. There's one coming up in less than 3 weeks. The ESIP Lab, which supports the development of innovative applied earth science technologies, and through 30+ community-driven virtual collaborations that span a wide range of topics. Some are more technical, like machine learning and cloud computing. Some are more application area-based, like agriculture and climate and disasters. And some are like this physical samples cluster. So we're very delighted again to be here and invite you to join in any of those collaborations.
Thank you, Megan. Before we get started, I do want to go over just a couple of little housekeeping items. We will be recording this presentation, and I will add the link to the recording on the RDA website following, well, once the recording is finished. Also, the slides will be uploaded to this page as well. Each presenter will leave 1 to 2 quick questions at the end of their talk. There's 4 presenters, and we'll also leave time at the end of the webinar for more in-depth questions. We would appreciate if you could type your questions into the Q&A part of your it's part of the Zoom panel, I guess you call it, control panel. And if you want your questions directed to a specific presenter, please put at their name so we know who to do. If for some reason we run out of time for the Q&A at the end, the questions will be recorded and we will get answers for them, and I'll send them to you in a Google Doc or a link to the Google Doc after the webinar. And again, we thank you all for attending, and I will now turn it over to our moderator, Esther. And Esther is one of the co-chairs of the RDA Physical Samples and Collections in Research Data Ecosystem Interest Group. Take it away, Esther. Thank you.
Thanks so much, Stephanie, and thanks so much to the RDA US for hosting this webinar today, which is the first webinar of the webinar series on physical samples. And we're thrilled to be jointly presenting this webinar series together with the RDA Physical Samples and Collections group from RDA and the ESIP Physical Sample Creation Cluster. But before I tell you more about these 2 groups, we would like to get a general idea of who you are. So I'm going to launch a quick poll about what your primary role is, and that will help our speakers to fully address the audience. So if you can quickly fill out the Zoom poll that was just launched, I'll give that a couple more seconds so everyone has a chance to fill it in. And I'll tell you more about the ESIP physical sample curation cluster, which was initiated in 2020, and it aims to enhance the discoverability, access, and use of physical sample collections. And the RDA physical samples and collections group was initiated in 2017 during the 10th RDA plenary. And the group aims to facilitate cross-domain exchange on key issues and recommended practices related to the digital representation of physical samples and collections. And so since these groups have very similar aims, we are now co-organizing this webinar series to increase the awareness of the importance of physical samples in research. And I will now end the polling, and I can hopefully also share the results. So it seems that we have a lot of librarian, data managers, and data stewards in the audience, but also researchers, students, a couple members of ICT departments, a funder or publisher, and some others. So it might be of interest to type in the chat how you define as other. I'm going to stop sharing those results, and I see that someone is asking about the slides, and I'm not sure if these have been, yes, I think we're now at the.
Oops, sorry.
Thank you. Yeah, if you could, yes, please keep it at speakers, because I am very excited about the 4 speakers that we have for the webinar today. So I'll briefly introduce them at the very start. So we first hear from Harald Kusch. Harald works as a data manager for the Cluster of Excellence Multiscale Bioimaging in the Department of Medical Informatics in Göttingen. Harald will present the institutional use case for electronic lab notebooks. Harald's use case is followed by the limitations of sample management that Robert has encountered in the past. And Robert will be focusing on a researcher perspective. Robert Kossen works on research data management and infrastructure in the Department of Medical Informatics in Göttingen. And before this, he obtained his PhD in sensory and motor neuroscience and became a certified project management associate. After this, we will hear from Rory McNeil, who is the CEO of ResearchSpace. Rory leads the ResearchSpace team, which is continuously implementing new ways of making RSpace a useful research data management tool that enables reproducibility and furthers fair data principles. Rory will introduce the functionalities that electronic lab notebooks provide, with a specific focus on RSpace and its connected approach. This general overview is then followed by a demonstration of the inventory model of RSpace by Vaida Plankiet. Vaida is an UX UI designer at ResearchSpace and the inventory user engagement lead. Vaida is focused on incorporating user feedback into the product to make the inventory intuitive and user-friendly. So while our presenters are speaking, please a reminder to add your questions into the Q&A section, preferably with the name of the speaker that you would like to have address your question. And we will have time for 1 or 2 short questions after the presentations by Harald and Robert, and then again after the presentation and the demo by Roy and Vaida. So I would now like to hand over the digital floor to Robert. And so please take it away. To Harald, actually, sorry.
No problem. So thanks very much for the nice introduction and welcome everybody from my perspective. I'm Harald Kusch from the Department of Medical Informatics in Göttingen, and I would tell you some comments and perspectives from our Cluster of Excellence MBC. That's my basic background, where I'm working as a data manager. And Robert will have the second part of the presentation. So about the scientific background, that you've got a feeling for the use case that I would like to present here. We are in life science, chemistry, and physics. So it's a broad range of different fields that we deal with. Most frequently, it's basic research questions that are addressed, but also ranging into translational research. And yeah, due to that broad context, broad scientific context, there's also a broad application of different kinds of samples. And I just put on that slide 4 examples. So we have a lot of animal models that are worked on, cell lines, antibodies, or plasmids. That's very frequent types of samples that we deal with. Next slide, please. So along with all these different backgrounds that we are dealing with as data managers in daily practice, there's also, regarding sample documentation, very heterogeneous aims. And that's, of course, also important for thinking about tools, processes, and managing sample documentation. So the first very prominent aim, of course, is the daily lab or even field work, where lab members or lab directors or admin staff in the lab needs to deal with samples and their documentation. A big part, at least here in Germany, is also a lot of authority reporting. Mice experiments, for example, are a prominent example. So there's a lot of sample documentation that needs to be done for authority reporting. Then there are 3 other levels that we as data managers also deal with, and that's at first research group wide internal management, so across a complete research group to manage samples, then also across consortia, which might be quite large with a lot of different lab groups, doing a joint internal management of sample documentation. And of course, as maybe everybody in the round here today, we are very interested and very enthusiastic about fair and open science approaches and try to make data as fair as possible, and also data regarding sample documentation. Next. Along with this heterogeneous background, there's of course a lot of challenges that we have to deal with. I cannot make a complete picture here, but some major aspects. The first one is heterogeneity, which is due to the different fields that we deal with. So samples can be of very, very different types, and we have to deal with all that in sample documentation. And it's also a lot of dynamics in the game. Highly dynamic sample management is daily work. So material is not always available. It's somehow gone. So every experiment has been carried out and the material is gone. So we have to track the material content, so to speak. And unfortunately, also in many instances, we have a high dynamic of team composition. So we have people joining a lab and people leaving a lab. And that's also something which is very challenging in sample documentation. And what makes it even more complicated, you all know that we have limited resources and on different levels. So maybe most prominent, the personnel is limited. But also, if we have enough personnel, if we are very enthusiastic about fair sample documentation, tools are not quite always available to do the right job. And we hopefully can show some approaches today how to improve the situation. And also processes regarding sample documentation might be very complicated and so might not be available in the labs because, again, personnel and tools are not readily available and there's a lot of work to do and to go. Next one. The practical approaches that we have in our labs that we have a glance on and can work together with, that's summarized on that slide here, and that's a kind of increasing complexity or professionality of sample documentation that we work with. So a lot of labs are managing samples on paper. They are working paper-based, and that's in the lowest organization form, decentralized, with different paper batches at different positions, maybe in the lab. But often this is centralized. There's maybe one folder somewhere where something around samples is centralized. A lot of other labs, maybe also in parallel, work with spreadsheets of different types like Word or Excel format. and also very common in labs for very important sample types. There are local databases available like FileMaker or Access databases. But what we clearly intend, and that's the plan also to show this today in this webinar, is centralized managing software. And there's a strong gradient between these examples that are listed here: electronic lab notebooks and lab information management systems. So both terms are used for different types of software, and we also apply a lot of these elements in our workflows as far as possible. Next one. And I just want to give you one example of our practical approaches, especially regarding fair sample data management. This is what you can also have a look at if you navigate to the URL at the bottom. So this is the research data platform built on the Minochi software, where you can have a look at all the publications that were derived of the cluster of excellence, which is of course a growing database, and you see a lot of documentation around the publication and associated data. And I want to draw your attention on the antibodies. They are very important and very costly sample material that is used along with the experiments. And you find a lot of information on these antibodies on the web page of the research data platform, also some quality information and the information where you can order that antibody. So this is just a very brief example now how we handle and share sample documentation with the scientific community. And this is the end of my part of the presentation. And now I think I hand over to Robert's part.
Yes, exactly. Thank you. So after Harold just gave you a bit of a broader overview and general overview about sample documentation, the requirements and the challenges, I'll try to do the same thing from a more lab perspective and tell you a little bit about how sample management, sample documentation, and the challenges therein are experienced by researchers on a more day-to-day basis. That is because, as Harold has already touched upon, collection, interaction, and usage of physical samples or materials is a fundamental part of many disciplines of the STEM fields. And depending on what disciplines one might work in specifically, these might include stuff like chemicals, model organisms or lab animals, especially transgenic ones, antibodies, electronic components, or many more that we do. And as there is such a high variation in the types and of physical samples and how they are used, there's also with it a high variation in what is the relevant and what is the necessary information in metadata for each of the physical samples. And with it, there's also a bit of variation in how are these informations cataloged or tracked, if they are even cataloged and tracked at all, which sadly is not always the case. One big issue that one encounters here in the day-to-day life is basically that there is a scattered information when it comes to physical samples. So the samples themselves that the researcher might work with are often only stored together with minimal information about the sample that makes it identifiable. So if you have a look at this little screenshot at the bottom of the slide here, there's a little excerpt from my past as a Drosophila neuroscientist. And you here can see just a screenshot of some Drosophila transgenic lines. And next to the genotype, you see in blue a little bit of text that is already an abbreviated version of a complete fly genotype. And you see that's already quite a big block of text that you would often simply not store and write on a little vial that only holds a couple of dozen flies. And this doesn't even include more extensive metadata information, such as raising temperature or special food conditions that might apply. So the complete information about a physical sample is often stored in a central document or repository, which I would call the best case in quotation marks, simply because it's the best case that's in use, actually. Or sadly, often this information is also stored in personal files of a researcher, which might not be easily available to another researcher. And this scattered information also extends to the documentation of experiments themselves, as oftentimes while performing an experiment, a researcher will simply not have the time to write down all the information in an extensive format. So they will use abbreviations, they will use certain stock numbers or similar information that allow them to store all the necessary information in the notebook, but not the complete one. And this, of course, these factors have a potential issues and implications for the reproduction and reuse of these experiments or the samples used therein. So at a very base level, the first thing that comes to mind in this case is that this can result in a limited reproducibility if, for whatever reason, certain staff might not be available. The person leaves the lab. For whatever reason, it might be really hard to completely reuse or reconstruct their experiments, as you have to parse through multiple instances of information, old lab notebooks, central repository, maybe certain documents or sheets of paper that they took with them to the experiment, all scattered over the place, only containing minimal information. So there can be quite a paper trail, sometimes a digital paper trail, trying to reconstruct the experiment. This, of course, gets only aggravated if we deal with the possible loss of certain physical documentation, even if it's just maybe a coffee stain on a lab notebook that makes a certain passage unreadable. Furthermore, this scattered information and improper traceability makes the sample exchange and replacement between researchers very difficult. So maybe I used up all my antibodies and I want to know who else uses this antibody so that I don't have to wait for the redelivery. Or maybe because of an unfortunate event, a Drosophila stockline that I used might have died. It would be very fruitful to see who else might have a copy of this stockline so that I don't have to spend money and time on getting a new one. And this in turn then leads to the idea of an event history for certain physical samples, because for certain physical samples, it might be really good to know who has worked with it and what was done with it. That might simply be because I'm trying out a new antibody for the first time, and maybe I want to ask my colleague who has already used it before, are there some tricks, are there special procedures I have to keep in mind when working with this? Or certain samples might have to be handled in a certain way. And it would be good to be able to reconstruct if they were always handled the proper way down the line before I started working with them, or after I might have given off the work to someone else. So these are all just the very slight insights into some of the issues that arise on a day-to-day basis. And just as a teaser of what comes up next, because I talked so much about problems, is that these are not insurmountable problems. And one very elegant solution that might at least alleviate some of them is a digital interaction, digital tracking of physical samples that directly works together with the day-to-day experimental notations and the electronic lab notebook that helps to keep everything more organized, metadata and information more complete. And I think this already brings me to the end of my part. At the end, we also, of course, have the obligatory slide that shows you that whether it be in research itself or in research assisting data management and infrastructures, no work is ever done by one person or a few persons, always a team effort.
Thank you so much, Robert, and also Harald. We have a question, so we're just going to do a quick question right now. We actually have multiple questions regarding persistent identifiers. Both Scout Calvert and Anita Bondrowski are asking about this. Anita noticed that you're using RRIDs, so these are resource identifiers for the antibodies. And she is wondering if you're asking people to register these. And Scout is wondering how widespread the adoption of these persistent identifiers is. So if you could please tell us something about that, and Harald first would be good.
Yes, I tried to make a start, and thanks for the question. I very much like that topic, of course, as a data manager of persistent identifiers, even though it's, especially for clinicians, quite far from daily practice, and it's also part of the truth. But that's exactly one part of the technical backbone that we are trying for years actually to implement here also in Göttingen, to use persistent identifiers wherever possible. And of course, there's a large variety up to now. You also see some on this last slide, the ORCID IDs or the ROR IDs for institutions, and we try to apply as much of them. But you're also addressing a very wounded point somehow. So data curation is in most parts our personal work. That's work of the data management managers. And of course, that's something we want to change, but it's really hard to change it, that the researchers themselves do it. However, what is really helping us is the increasing efforts that journalists do actually to enforce authors to deliver these kind of accession numbers as well, which is of course also expandable and optimizable, but it's happening, and this is what we can easily use for data curation.
Thanks so much for that answer. Robert, do you maybe have something to add? Or do we want to discuss the adoption of persistent identifiers at the end if there's still questions?
I think Harold covered the most important part for this.
Okay. In that case, I would like to thank you again for the presentations. We do have to go to the next presentation, which will be by Roy and by Vaida from RSpace. So if Roy can share the slides and turn his camera on. Yes, that's perfectly visible. We cannot hear you yet or see you though.
Hello, everyone. I'm just trying to get my my screen back here. Trying to.
It was in presenter mode a second ago.
Yeah, it's when I switch to. Anyway, you won't be able to see me, so but let me press on. Oh, here we go. Start video. Ah, there we go. Great, fantastic. Okay. Hi, everyone. Thank you to everyone for organizing this, and thank you, Esther. So we're going to take a step back, and before we pass over to Maida, who's going to illustrate how RSpace and our new inventory module are able to deal with some of the issues that Robert and Harold mentioned. We're going to take a step back and look at RSpace, which is the context for the inventory module. So RSpace is an ELN, or an electronic research notebook, which is a growing term, that's used to collect, organize, manage, and share data during the active research phase. It's built around academic workflows, so RSpace is one which is particularly designed for academic context. It serves as a centralized data hub. One of its key factors is that it integrates with other tools that you're already using as a researcher. Importantly, it's intended as a conduit, not a silo, and it connects data management during the planning phase, the active data phase, and then the data archiving and storage phase. And it's completely focused on enhancing reproducibility and verification of data. So to bring those points home, I'm going to look at this, take a look at this graphic. There's quite a lot going on. So if you imagine in the middle, there's RSpace, and that's what you would expect, all the things that you would expect an electronic lab or an electronic research notebook to be able to do in terms of structuring data, collecting data together, sharing data with your colleagues, having an audit trail, being able to sign witness documents, et cetera, et cetera, et cetera. But the focus here, what makes RSpace distinctive is its focus on the ecosystem, and it's the ecosystem which enables particularly the enhancement of reproducibility. So you can see that there are a lot of things connected to RSpace here. I'm now going to delve in and talk about briefly about each of them categorically in turn. So first of all, RSpace is connected with lots of things which allow you to incorporate data from external resources, either by importing into RSpace or by linking to them. And this includes things like data storage devices, be they institutional or commercial offerings, like Box and Dropbox, Google Drive, OneDrive, and also things like OwnCloud, which I know are the backbone of many of the European universities' data storage devices. It also includes things like resources data, which is captured in tools, both things like Office 365, protocols, IO being an example, as well as specialized kinds of data like the PIRET colony management system. That was in one of Robert's slides of one of the kinds of things which are captured. You can also incorporate data from collaborative tools like Slack and Teams, you can dump things from conversations into RSpace. And we're beginning to look at putting voice data in as well. Secondly, once the data is in RSpace, it can then be exported to data repositories like Dataverse and Figshare, with which we have bespoke integrations. So it's possible to export data in a wide range of formats, including ORCID IDs, to Dataverse and Figshare, so that not only data in RSpace, but data from this broader ecosystem can be associated together and then put in Dataverse and Figshare for querying, for reuse and attempts at reproducibility. This is slightly off topic today, but one of our newer things we're doing, which I think will be interesting to some of the research data managers. We're working with the folks at the California Digital Library on an integration with both DMP Tool and DMP Online, so that it will be possible to associate DMPs with the active data that's research going on in RSpace, and then again, put both of them into data repositories. And this is in an effort to make the DMPs more of a living document and less of something that just gets used and discarded. Today, of course, the focus is on sample management. So Vida is going to be talking about RS Inventory, our new sample management system, how you can associate sample data with experimental data in RSpace, and then export that to a repository, which we are pretty confident is going to allow a step jump in overall experimental reproducibility. And in particular, it addresses a number of the questions and issues that Robert and Harold have clearly identified. So thank you very much. With that, I will stop and pass over to Vida.
Awesome, thanks, Rory. I'm just gonna share my screen. Can everyone see an inventory system? Amazing. Yeah, so thanks, Rory. So as Rory mentioned, RSpace itself on its own is really good at enabling reproducibility in a uniquely powerful way, but it is really the sample and inventory management system that we want to showcase today, and it's a deep integration with the ELN because it really takes reproducibility to a whole new level. So I'll start by giving you a brief overview of inventory, and then I'll showcase the ELN integration in more depth. So here we have an example inventory with some test samples. So I'm going to show how inventory really is highly customizable, which makes it very powerful, as it can then be extended and configured so that it would be suited for the needs of your community. So here I have an example sample. It has some default properties like an image, a name, an expiry date, a description, and some tags. But what's really important here is that I'm actually creating the sample from a custom antibody template. And so when creating this template, I've defined which fields I want to associate with it. So this is an antibody template. So I have information, a text field, some information about the antigen, peptide. I have some fields that are checkbox fields that I can select. I have defined a field that's a radio field, so I can only pick one option out of a few. And in this way, I can ensure that I'm creating sample types that are really consistent and allow me to record data in a consistent way. Another thing to mention is here I've created a custom field for an identifier, the IGSN, and I can just paste in a link to that resource's unique ID. Thanks to Jens Klump from Cyro for suggesting adding custom identifiers. And as mentioned in chat and in the questions, this seems to be a really, really big topic, and that's something we really want to implement. So we'd love to engage with researchers to figure out which identifier schemes we should support by default, for example, by providing just like a drop-down list that you can select from. Just a really easy to use way to link all of the inventory items with their actual persistent identifiers. So especially from the geo and non-bio community. So please get in touch with us. We'd love to hear from you. So a little bit more about sample templates. Let me just scroll back up and open the information on this template. Here's again a recap of all of the fields I've created. Let me click on the template, and here let me edit it. So if I scroll down, I can see here that I can really customize things deeply. So maybe these subsamples I'm working on actually are not called subsamples, but I want to call them pieces or portions or units, and I want that to be reflected throughout the inventory system. You can do that. Maybe the quantity I'm working on is a volume and it's liters, or maybe it's mass, or maybe it's completely unitless. And again, I want that to be reflected in the system and for the sample type. And I can just do that. And here are some fields examples. We can set default values. So that's really nifty, and again, quite easy to use. Now, you might want to create a field for, let's say, safety fields to link to a PDF of the sample information regarding safety, or have required fields for reports. And again, you can all do that. That's supported by the system. Now, let me quickly go back to my sample. Yeah, we don't want to edit that. And as you can see, as I'm navigating through a little bit here, it is quite, oh, let me just check if, oh, okay, there's just some notifications in chat. Okay, everything's working. So we have quite a modern, simple interface because we really want to make sure that we're showcasing all of the important information without overwhelming you with information. So we're continuously reworking the interface to be minimalistic, but still provide a lot of power. One thing is, for example, let me scroll down, let me select the subsample. When I'm working with samples, I want to know right away, okay, where is this subsample? Well, that's straightforward. You just got a breadcrumb link here at the top, and this shows me that this antibody is in a tube grid, which is in a large box, which is in a container rack. And now I can click on those breadcrumbs to actually load in that specific container and have a look. Okay, this is a grid container. I've got some items. I can hover over the info icons to see core information about that sample. Maybe I want to go to its parent container. Okay, well, let's click on large box here, see the parent container. This is an example of a visual container. So because this container has a bit of a weird shape, maybe I've decided, okay, I want the inventory system to really replicate what the physical container looks like. So I'm going to take a picture and I'm going to set a few locations in the system. Okay, this first row can only handle 2 locations. This one can only handle 1, and so on. And then I can move the specific aliquots or containers into this bigger container. Again, really enhancing the visuality aspect of an inventory system, which is really important. Another aspect is that not only do we have different container types, we also have different ways of viewing them. So for example, maybe visual container view is not exactly what I want right now. I might want a card view where I see images and then core information about each item. Or let's say I'm navigating containers. And when I'm navigating containers, that's a bit like navigating a file hierarchy, right? So I might want to switch to tree view. And here I might want to click on items and then expand into different containers to be able to see and find the exact items I want. So again, not only is there real flexibility in how you name and how you create items, but there's also a lot of flexibility in how you might want to see and interact with items, which is really, really nice. So that was very quick, but now let's switch over to specifically the ELN. So here we've got an ELN document, just pretty classic. You've got some fields, we've got some tables, some entries. Now notice on the right-hand side here, we've got little inventory icons. Now, if you click on one of these icons, you'll access the list of materials. So the ELN integration is really about enhancing reproducibility by making it possible to include reliable data about the samples used in the experimental records. So let me open the list of materials here. I can click on add items to add some items in a very specific interface that we're just reusing as well in inventory. It's very straightforward and powerful search, which allows you to really find the items you need. And here is a table of the subsamples that I've pulled in for this specific experiment. So it's got all of the essential information you need. So let's hover over the info buttons. We get some essential information. We have some quantities. I'll explain this in a little bit. We've got the location so that you can just click on the specific location of an item and be able to access that container to figure out where that exact item is. and be able to find it. And if the item is moved in inventory, this location column will update. So you'll always have the most up-to-date location. Then the global ID, this is very important. This is the RSpace persistent and unique identifier for that item. You might have noticed as I was navigating around the interface here, we've got this column here in list view, always of the global ID of that item, whether we're viewing containers, templates, or samples. And we've also got this global ID on the top right here. So it is created and associated with an item right when we first create that item. And it is also included in the ELN integration to make sure that you are always linking in a very unique way to that specific item inside of inventory. So actually it's clickable. So if I click on it, I just open that resource in inventory and can access all of its properties. But it also means that we're always keeping inventory and the ELN and the list of materials in sync, because we're not just kind of importing or copy pasting this information from inventory, we're dynamically linking to it. And that is very important in highly dynamic labs where, you know, the location might change and you need the location column to update. You know, the name or maybe the core information might change. You need that all to be updated inside of the ELN as well. Now, when you're working with samples, one common workflow is that you want your experimental record to accurately portray how much you've used of a specific item. So that is something you can do just simply by editing the consumed quantity of an item. Here I'm setting it to 3 milliliters, and you can see automatically the inventory quantity is reduced to reflect the fact that, you know, you've had a reduction in the quantity of your sample. And this specific quantity is automatically synced with inventory. So if you're browsing inventory, you'll be able to see, okay, I'm running low on this specific sample. Let me make sure to, you know, restock. And if someone in inventory edits that sample's quantity, you'll see it in your list of materials as, oh, okay, I'm not having enough for this experiment. I'm going to have to do something about it. So it's a very powerful way of making sure that everything is kept consistent and in sync, and that you're always handling the most up-to-date data for your research. Now, let me show you a few other features. Let's say, you know, you want a print or a PDF version of your document. That's super easy. You can just print. And when printing and having list of materials attached to your experiment, they will just be dynamically added to the PDF. So you'll have nicely formatted tables, exactly like in the panel, if you wanted to print out the contents alongside the actual experimental record. Now, something else you might want to do is, as we were mentioning repositories, you know, you're finished with your experiment, you're quite happy with how everything went, and you want to share your research with the world, and you might want to export to Dataverse or maybe Figshare. So I'll quickly demo that. We have an export button here for the experiment. You can generate a file of like a bundle of web files. You can generate an XML bundle, which is great for re-importing into RSpace or for archival. So all the data associated with your experiment. A PDF file, a doc file. I'm going to choose a PDF file, and I'm going to choose to export to a repository. So I can choose some details on what I want to actually include. So do I want to include the comments made to this document? Maybe I've collaborated with different people and we've made a lot of comments on it, different PDF formats. Now, this is the interesting part. I've already set up a Dataverse integration. I mean, I know a lot of us are in this event are research data managers, so you'll know what Dataverse is, but just to quickly recap, the Dataverse project is a data repository specifically focused on open science. So you can make your research data available to others and make it more easy to replicate research, as well as make your own research more replicable, and your work can easily be cited, explored, and analyzed. So it's really pretty nifty. So here, let me write test RDA exports. Let's say this is a test description, just a bit of a typo here. Let me say this is medicine, health, and life sciences, and it's a draft. You can add some authors based on who contributed to this research. I'm just going to click on export. And again, integrations are really the strength of RSpace because it minimizes the amount of work you need to do for very common workflows. So here we can see a notification. Cool. Let's open this link for the Dataverse export. And here we go. We've got our export with all of the information already saved into it. And we can open the PDF. And as we can see here, again, we have all of the information for the experiment that's been recorded in it. But most importantly, all of the item global IDs are also recorded in the export, the type, whether it's a subsample or container, the name, how much of the actual sample we've used in that experiment. So as a result, as soon as we create an item in inventory, it has that global ID attached to it. And then when we are using an item in the ELN integration, we still have that global ID as a way to access that resource. And then after exporting, we still retain all of the sample information embedded within the experiment to make sure that it never gets lost. So that was a very, very quick overview of RSpace inventory. And it's focused on being customizable, intuitive, and then also tightly integrated with the ELN, which enables you to associate sample information with the experimental records and maintain this information in exports of your research, enhancing reproducibility. So we have plans for further ELN inventory integration to make the system even more powerful. This is just phase one. In the next phases, we'd like to make sure that on the sample page in inventory, we maintain links to the experiments in which a sample has been used. So basically the opposite of a list of materials, have like a list of experiments. And also we want to record sample provenance so that you can store links to the parent sample and child sample and the other way around. And we also will generate audit trail entries for when samples are associated with experiments to better keep track of how a sample has been used. So RSpace inventory is having its production release on September the 8th, but an alpha test version is available already, and it is updated regularly with the newest features as we move towards the production release. This open development enables us to really take in feedback from researchers and has been really invaluable. So if you'd like to check it out, I think in chat, Esther will pop in the link to our RSpace inventory hub, as well as my email, and we'd really love to engage with you and hear from you. The inventory hub also has a detailed ELN integration video from our latest release if you want to see more of the actual details of how it works and the features included alongside all of the documentation for that. So thank you so much for your time. And yeah, please feel free to ask any questions about all of the things we've covered today, and I'm looking forward to answering them.
Thanks so much, Vaida, and also Rory. We have a lot of questions actually. And our first question was from Flora Dana. Can you tell us a little bit more about collaboration? Who can edit the inventory? Who can make a template? If multiple users edit the inventory at the same time, do you have conflicts?
Yeah, I can answer that. So right now, for the issue of if two people try to edit the same item, we have edit locks in place so that if one user starts editing an item, all the other people are temporarily unable to edit the item. And then once the user saves or after a certain amount of inactivity, that item is then released to the other users. So that's not a problem. When it comes to permissions, I'll just pop an article in the chat. We've developed some basic permissions. So far what's really supported is more of an open lab setup. So where all of the users in a specific lab group can share and view each other's items, but then you can also transfer your items to another user outside your lab group perhaps. But it's quite open for now, but we obviously can easily implement similar controls to what we're doing for the RSpace side, where we have PIs and lab admins that do administration, but then we also have lab members that mostly can work on their own items perhaps, but we also have communities and system administrators that can also do some more administrative tasks on top of that.
Thanks so much for that answer. I hope it's addressed the question. And we also had a question about the exporting of the information. Does it also include a region table with the appropriate persistent identifiers?
Rory, would you have some feedback on that one?
Yes. Yeah. So I think not automatically. I mean, actually, I hope Harold or Robert might comment after I comment. But I think the reagent table would be something that the researcher, the person doing the research, would add themselves in the ELN side, and then the sample information would be associated with that. So it doesn't happen kind of automatically or magically, but certainly if that was a feature of the write-up of the research, then the answer would be yes. But Harold and Robert, I'm curious, what would you say about that?
So maybe I make a start. So we are really happy about being part of the construction actually of that inventory, and what we are trying to do. And this is also one of the outputs, this kind of perspective that we showed today, that we are closely working together with the scientists to observe their workflows and find out what they really need. Of course, we as data management managers have a lot of funny ideas what we could do. And this is potentially also true for software companies, but sometimes it's not really what people need in the lab. And the ways of reporting can have very different aims and really need to be designed to the specific use cases that the researchers have. I mentioned, for example, authority reports are totally different from lab documentation for your own lab experiments. And yeah, but any type of automation in that process, of course, is very helpful and useful.
Maybe Robert, if you have anything to add, feel free. If not, I am going to move to a next question, or actually I'm going to ask first a question to the both of you again, Harold and Robert. Would it help if your institution, Göttingen, has a research data management policy that actually encourages the use of electronic lab notebooks or data sharing, or does your institution already have this type of policy?
It's also a very good point. So having support by the institution is a very good motivation, of course, for encouraging all these kind of processes that we are discussing today. And fortunately, we have a lot of that already. Not enough, that's also true, but we have something. And I'd first like to mention the eResearch Alliance here in Göttingen, which is very active in promoting all kinds of data management practices and open science. And I'm also part in one of the committees there and try to build the bridge between medicine and cross discipline discussion. So there's a lot going on, a lot of support which is needed and useful, but it's never enough, I fear.
Never enough indeed. Thanks so much for that answer. Robert, did you want to add?
No, all good.
Thanks. Then we had a question on how uncertainties are being handled in RSpace, and perhaps Vida or Roy would like to add to that. So for example, acceptance tolerance for recipe ingredients, how would that work?
Vida, do you want to comment on that?
Yeah. So I guess this depends on your specific use case. In terms of the custom fields, you can always fill in whatever values you need in terms of having a number field or a text field. So you could decide to type in a tolerance there. When it comes to the actual quantities of items in inventory, we do have a feature where, let's say, if your actual consumed quantity is larger than what you had recorded in your inventory quantity, there's a way to manually edit the inventory and quantity as separately from the consumed quantity to ensure that your actual experimental record is always accurate, and you can always modify the inventory quantity afterwards to be most accurate. Hopefully that answers the question.
I hope so as well. If not, please type in chat. We have time for one more quick question if anyone has one, or if I forgot to address your question in either the chat or the Q&A. And while we wait for that, I'm just going to ask Vida again to highlight if, for example, we want to have the RRIDs implemented in RSpace. How can we just email you, and how would that work?
Yeah, that's correct. If you could just email me and get in touch if you want to either, type in some ideas, if you want to just schedule a short chat, that would be really great. I saw there was some feedback in chat already that we'll definitely also implement. So yeah, just to recap, like the IGSN is just an example of just creating a custom field with a specific value. You can create custom fields for any kind of value you want. And what we're trying to aim to do is to make it so it's really straightforward. So for example, you can select a specific type of identifier from a dropdown and just paste in the ID, and that will generate the link to the item. So you don't need to do that manually. That kind of time-saving feature.
Esther, can I just comment briefly as well? So yes, thanks. Just further to what Vida said, this is like a dream come true, because this has been a very much community-driven interactive project, thanks to people like Robert and Harold, but also many other people. And persistent identifiers have in the past fairly recently become very much to the fore. So what we were really hoping was that one of our objectives in today's session was to meet people, engage with people who can give us feedback generally, but in particular on persistent identifiers, how to incorporate them, how to access them, workflows, different organizations that are out there. So absolutely, if any of you are willing to engage with us, we will be thrilled if you'd email Vida so that we could learn from you and start a conversation.
Excellent. Thanks so much for that call to action. I hope people will pick that up. And with that, we're running out of time. So I would like to thank all the speakers for their answers. And I would like to hand over the floor back to Stephanie.
Oh, thank you, Esther. Let me start my video. Okay, what a wonderful presentation. Thanks to all of the presenters and for a very informative presentation. I especially want to thank Megan Underwood for supporting us technically, and Esther for all your help. Thanks so much. And to the audience for all your wonderful questions. If there's other questions we haven't gotten to, we'll send you answers. Wanted to bring to your attention the next webinar that's coming up on August 10th. And that will be persistent identifiers, RRIDs, a way to track samples through the scientific literature by Anita Bandrowski. And she's been adding questions and things during this call. So mark your calendar for that. Again, if you have not joined RDA, we would love for you to be a part of this community. And the web address is on the screen. And I want to thank the RDA US, and ESIP and all of you for joining us today. Have a great day. Thanks for coming. Best wishes to all of you. Bye.